So for our lab 3, exercise 24, we're going to be talking about prokaryotes. So here are some very general characteristics that we need to know about while we are studying prokaryotes. The first being that they are unicellular organisms. So that means there is just one single cell. They do not have a membrane-bound nucleus or organelles. So they do not have that endoplasmic reticulum. They don't have Golgi apparatus or any of that. They have instead a nucleoid region where their DNA is at. Some will also have plasmids, which are just little chunks of DNA. And they do have ribosomes, but they're not the same as eukaryote ribosomes. These ribosomes do not have membranes. They're surrounded by a phospholipid bilayer cell membrane. That's what is keeping that unicellular organism intact. They have what is called a cell wall, and it's made out of a protein complex called peptidoglycan. Some prokaryotes will have a lipopolysaccharide layer, also called an LPS layer. And then some will even have a slimy or waxy capsule coat that helps keep them like stuck in place or stuck to a surface. To reproduce, prokaryotes will do binary fission. That's where they'll duplicate and then pinch in half. It's a type of asexual reproduction. And then there's also conjugation, where they transfer DNA from one cell to another, and that's considered a sexual reproductive method. And then, of course, some are pathogenic, or that means causes disease. If we look at our taxonomy, we know that we have the domain Archaea, and under that domain, we have kingdom Archaea. Now, there are a lot more taxonomic classifications, but for everything that we will do in our lab, as long as you know that you have the domain Archaea and kingdom Archaea, as well as the domain Bacteria and kingdom Bacteria, then you will be good. In the domain Archaea, kingdom Archaea, those prokaryotes are often ones that can live in extreme and stressful environments, where our domain and kingdom bacteria are much more common that you find every day around all surfaces. Let's first look at the bacterial cell morphology. So the first kind of cell shape that we see is called coccus, and that is a circle or sphere shaped. We can have these sphere-shaped coccus bacteria in groupings, and we give them special names. If there's a pair of cocci bacteria, we call it diplococcus. If there is a group of 4 cocci bacteria, we call it a tetrad. If there's a cluster of cocci bacteria, we call it staphylococcus. And you can think of a staph infection as being something that is caused by a staphylococcus morphology of bacteria. And then streptococcus is a chain. And I like to think of strep throat, and your throat is a long tube. So a long chain of cocci bacteria is sort of able to be thought of like strep throat, so streptococcus. Then you've got bacillus, which are our rod-shaped bacteria. They also sort of look like a Tic Tac or a Mike and Ike candy, sort of ovally shaped. Then spirillum, that is going to be this fixed spiral shape, sort of like a slinky or like a piece of confetti. When we look at our gram-positive and gram-negative bacteria, what we're really looking at is what color are these bacteria under the microscope when we stain them with a special process called gram staining. If the bacteria has a very thick peptidoglycan cell wall, that gram-positive bacteria will look purple or blue because it's going to hold on to that pigment in that peptidoglycan cell wall. Gram-negative bacteria have a thinner peptidoglycan cell wall, and they have a much thicker outer covering of that lippy polysaccharide layer. That lippy polysaccharide layer does not hold on to that purple dye. It will actually take up a pink or red dye. So gram-negative bacteria look pink or red. How do we do gram staining? It's actually pretty simple. We would take our bacteria and put it on the slide, and then we would apply a dye called crystal violet. And crystal violet is purple. The second step is that we apply iodine. It's sometimes called a mordant, and it helps that purple dye stick to that peptidoglycan. Next, we would wash off the staining with alcohol. We call that the decolorization step. And then we would apply safranin, which is a counter stain. It's lighter than the purple dye, so that any bacteria that didn't hold on to the purple dye, it's able to be covered over with pink. And so then we will see that our gram-positive bacteria will look purple or blue, or our gram-negative bacteria will look pink or red. So again, here's how this works. The gram-positive have this thick cell wall and no LPS layer. So when you stain it, the crystal violet makes it look blue or purple. Gram-negative bacteria have a thin peptidoglycan cell wall with a lippy polysaccharide layer. The crystal violet doesn't stick well to that layer and gets washed away with the alcohol wash. And then when we stain it with safranin, it turns pink or red. On the D2L for you are several videos showing you different stained bacteria under the microscope. Unfortunately, you wouldn't be able to see it with your normal eye, and we can't use the microscopes like you would be doing in the lab. So those videos will show you what you would have seen and the difference that you can tell under the microscope. In our prokaryotes, we also want to talk about cyanobacteria. And from lecture, you have found out that cyanobacteria are responsible for creating oxygen in the early Earth's atmosphere. Cyanobacteria are photosynthetic, so by that process, they make oxygen. They are also called blue-green algae, but it's important to know that cyanobacteria are not a type of algae. They are prokaryotic, so they cannot be the eukaryotic algae that we will discuss in our next lab. Also, they're not always blue or green. They can have many other pigments. There are many other pigments even in our plants that can make them turn yellow, brown, or red. And this is one reason why it's important to have taxonomic and scientific naming instead of just calling it what it looks like, because it avoids confusion and misidentification. A very important process that cyanobacteria do in our world today is something called nitrogen fixation. Some bacteria and many cyanobacteria are able to do this process. They're able to take nitrogen that is in our air and our atmosphere and to make it into ammonia, NH3. And plants cannot do this on their own. So they need the bacteria and cyanobacteria to do this so that they can take up that ammonia and use it to make proteins and nucleic acids. No life on Earth would be possible without this activity. One species that we have a video of on the D2L and that you would look at if you were in lab is called Rhizobidium, and it's going to live with plants in their roots. And they form these little nodules or sort of little ball shapes on the roots. And they're able to live together because the rhizobidium is able to give the plant ammonia to make proteins and nucleic acids. And then the rhizobidium is given sugar from the plant. Very briefly, we want to talk about bacterial cultures. In order to study bacteria in the lab, we have to culture or grow them, and we do that in what we call nutrient media or agar. With this agar, the bacteria are able to reproduce quickly, and then it's because that agar gives them food and water. When we look at the bacteria cultures on a plate, like the picture on this slide, we're not seeing individual bacteria, but we're actually seeing millions of cells that make visible colonies. Each bacterial species always forms a colony that looks the same way. So if it is a pink and fuzzy looking thing, it will always be that way. Or if it's this green and bumpy, it will always be that way. So that is one way that we are able to test for and ID different species when we're making our cultures. So what are you going to be doing in your lab? Well, you're going to be looking at different bacterial cell morphology. There are prepared slides in the form of videos and pictures on the D2L. And so you're going to want to be able to draw a sphere-shaped bacteria, a rod-shaped bacteria, and a spiral-shaped bacteria, and then be able to give the names of that morphology, whether it's coccus, bacillus, or spirillum. You'll also want to be able to look at it and see if it's pink or red or blue and purple, and tell me if it's gram-negative or gram-positive. You'll also be able to look at some videos of cyanobacteria, specifically Nostoc, Oscillatoria, Glocapsa, and there is another one, Anabaena. In all of those, you're going to want to find what we call the heterocyst. That's the area where the nitrogen fixation is taking place, and it's the larger cell that you'll see in the little colony of cyanobacteria. That's really it for this lab. So hopefully that is enough information to get you started. Of course, if you have any questions or concerns as you're going through this, just let me know. I'm happy to help. Have a great day.